point, I'm going to be talking to you today about horizontal gene transfer and its effect on genome modularity and evolvability. So, as usual, I like to start with not actually advancing the slides. All right. One of the things we like to start with is the acknowledgments. I should point out this is all a computational system that was largely built by Charles Zofria there. The specific commands that I need for the work that I'm doing were mostly programmed by Rose over there. My programming is statistical. I am not a computer programmer. I rely on these people. Also various funding sources. So, as a bit of a warning, eukaryotes are weird. They're a minority of the population in terms of life, even though they're what a lot of us tend to think about when we're thinking about living things. And that's because we have this bias due to the fact that they are the large multicellular organisms. They're the things we see most of the time, even though they're a very small subset of living things. This sort of tree of life is something many of us have seen, many of us have been taught with. This actually exaggerates the importance of eukaryotes. This is a slightly more realistic sampling of life. You'll notice that the vast majority of it is in blue, which is bacterial, and that's even leaving off the viruses, which are even smaller and even more diverse. In addition to being a little bit strange in terms of their physical proportions, eukaryotes are also unusual in the way they organize their genes. So if I'm to plot out what I'm going to pretend is a typical eukaryote, and in this case that's a human, if we look at the haploid set of chromosomes, this is the set of chromosomes, and I want to take a look at one particular genetic complex. So for instance, the succinate dehydrogenase complex, which is made up of 4 different gene products. It's an important part of central metabolism. If I plot where those are, well, the first one's here on chromosome 1, and then the next one's over on chromosome 5, and the third one's back on chromosome 1, and the fourth one is out on chromosome 11. And this is actually a pretty typical pattern of how genes are distributed in eukaryotic systems. So they're often very highly dispersed across the whole genome, even when they're part of the same complex. On the other hand, things are very different in prokaryotic systems. So this is the succinate dehydrogenase operon from one particular microbe. You'll notice that those 3 genes are directly next to each other. They're actually under control of a single promoter. So bacterial genomes are arranged in a very different way compared to eukaryotic ones. And there are a couple of different possible reasons for that. But one of the major reasons that's been brought up is the possibility that this is being driven by horizontal gene transfer. So there are a couple of different mechanisms of genetic transfer. There's the vertical system that we all tend to think about, where organisms get their genetic material from their immediate parent or parents, depending on whether you're asexual or sexual. So you get this transmission of genetic material down through a family tree, and so it creates this horizontal path. But that's not the only way that organisms can pick up their genetic material. You also have the option of horizontal gene transfer, where there's some DNA out in the environment which somehow gets into the cell, and there are a lot of different routes of that. But sometimes that actually gets incorporated into the host chromosome, and so this individual, most of its genetic material came from its immediate parent, but there's some portion of it that came from someone else in the environment. And horizontal gene transfer has the possibility of leading directly to modularity. So some random mutation makes this genome slightly more modular by bringing the pieces of it into separate places, or bringing those separate pieces that are part of an overall pathway closer together to each other. This more modular genome is more likely to spread this genetic material through horizontal gene transfer, because you've got these separate components that are using different portions of the genome, and if the genome manages to place these different components close to each other, a single transfer event can bring in all of the necessary components for a particular function. And this can lead to a selection for modularity. The problem is, this is really hard to test empirically. It's very easy for us to do certain types of empirical tests. It's not so easy for us to just disallow entire categories of mutation. And you might need very many generations to see an impact of this. And so to get around some of these problems, I've actually turned to a computational system to do this work. So this is work done actually with the research version of Vita. I have here a screenshot from the educational version because it has a nice graphical user interface, and that makes for pretty pictures and command line programming. What's going on here is that each one of these boxes is its own self-replicating computer program. But this replication isn't perfect. The user gets to define a mutation rate, a probability of miscopying one of these instructions for another. Because you have this self-replication that is error-prone, you have mutations as a source of variation, you have heredity, you have natural selection because these guys are always competing for space in the world. They're often competing for computational resources. They do certain tasks, we reward them by letting them execute their code faster, and therefore they reproduce faster. So we have all of the necessary components for evolution by natural selection. This is actually an instance of evolution rather than just assimilation. I don't have time to go into the details of everything that goes on in the system. At the moment, I can tell you essentially that this is a haploid system, that we are able to do things that more closely approximate bacterial work than they do non-bacterial work, but it's an approximation anyway. This is not a direct mapping of any individual biological system. This is an overall concept. So, the overall experiment that we're talking about here is that we set up two basic treatments. In one of these treatments, we allow horizontal gene transfer. So when organisms die, their genomes are fragmented. Those fragments exist as resources within the particular cell in the world that the organism was living. And so most of the time, those fragments are totally useless, but when we allow the possibility of horizontal gene transfer, and I need to get back to the next slide, when that is a possibility, there's an instruction that they can execute that will pick up one of these fragments from the same space in the world. There's a certain probability that when doing so, they'll degrade it and turn it into energy and be able to execute their code faster. There's also a probability that it will undergo homologous recombination within the host genome. So this is essentially akin to the concept of natural colonies. That they're taking in this genetic material as a possible energy source. There's a certain possibility that it will result in genetic transformation of the system. In both of these two treatments, we have environmental cycles. These logic tasks that I referred to before, in one of the two environmental states, they're being rewarded, and the other one, they're actively being punished. So organisms take a hit if they do this task. This is because in previous work with the same authors, we've shown that this cycling of environments is really important for getting horizontal gene transfer up and off the ground. So it actually goes into the system. We ran through 200 updates for each of these. Updates are a computational timescale. They are smaller than generations. The exact degree to which they are smaller than generations depends on how fit everything is in the system. But so this 500 update cycle is roughly 30 generations, but it's not consistently the same on every single run. We measured both the evolvability of the system, and that is how many different tasks the organisms have managed to acquire, and the modularity, so essentially a measurement of the number of tasks that each individual cycle codes for, normalized by the total number of tasks that the organism is doing. Because if you don't do that normalization, an organism that has more tasks is going to, by default, have more tasks encoded for each individual site. So you have to do normalization to get a fair comparison. So, what do we actually find from this? Well, the first one we find is that horizontal gene transfer does actually increase the evolvability. So the environment that we allowed horizontal gene transfer for is the one on the left in the light blue. The one where we didn't allow it is on the right, and it's in dark red. Since this is categorical data, it's easier to just plot out all of the individual points for you. I can also show you stats that is, in fact, a statistically significant difference. We had an increase of about 37% in the number of tasks that an organism is doing at the end of this run. If horizontal gene transfer was allowed, then if it wasn't, this is a highly statistically significant result. We also can show you that horizontal gene transfer does lead to a slight increase in the number of tasks encoded within any individual site. And this, again, the one with horizontal gene transfer is the light blue on the left. And so that is actually a slightly higher number of tasks per site. And that does also turn out to be statistically significant. But this increase is substantially less than you would expect just due to the increase in the number of tasks being performed, which indicates that when we actually normalize for the number of tasks being performed in the organism, we find that horizontal gene transfer does lead to more modular genomes. they are doing a better job of separating these tasks into different instruction sites within the genome than you would expect simply based on the number of tasks that they're doing. And again, this is statistically significant. And all of these significance values remain even when we correct for biases. So our overall set of results from this set of experiments is that horizontal gene transfer does lead to more tasks being done, which is a measurement of higher evolvability in the system. And horizontal gene transfer increases the number of tasks per site, but by less than the increase in the number of tasks, which means that it's actually resulting in a more modular pattern. Now, that being said, this is just one particular measurement of modularity. We're looking at how many tasks any individual site is involved in performing. There are other measurements of modularity as well, such as how compact those sites are together and how discrete chunks they are. We are in the process of doing those measurements, but I don't have the data for them yet, so I can't talk to them. And other than that, I am happy to take some questions. Yeah.
That was nice. Thank you, Mike. And I'm wondering what I can take from this were I to be motivated to go back into the lab or look at genomes. What are the hallmarks here that I might, or are there hallmarks here at the level of the genome that, or at other levels that I could take to the bench and look in the genome of E. coli or my favorite organism?
So if I'm understanding you correctly, your question is about what can we take from these findings for wet lab research that we would want to conduct?
Yeah.
And that's a good question and not something I've actually thought about. I was trying to get at this to address this long-standing question of why we even have this higher modularity in bacteria. And so we were looking for, is there a way to get empirical evidence in favor of one of these hypotheses?
Yeah, and that I really like. And I'm just wondering whether along the way you can see certain features of the genomes where you allow HGT that we should be looking for in DNA sequence data.
So I will have to talk to you about that a little more in detail afterwards. So we have another set of work showing that the cycling environments are also really important for promoting modularity themselves, but that's a whole additional talk beyond what I have time for today. But I can talk to you about that in one of the breaks.
How big are the chunks that you horizontally transfer? Like, are they X number of instructions? Is it?
So what we've done is we fragment the genome. So the question is how big the chunks are that are going through this horizontal gene transfer process. And we fragment the genome in a Poisson distribution, which our mean size of chunk in this set of runs, I believe, was about 15 instructions long. When we look for homologous recombination, we mandate that they had at least 3 instructions at the start that properly matches something, and 3 at the end that properly match. And the spacing in between can be as little as half the size of the thing they're replacing, or as much as twice the size they're replacing.
Okay, that was my second.
These 3 individual instructions with an alphabet size of about 26 is about the same degree of specificity as you would get within a DNA system of requiring about 8 base pairs for getting your match on the ground. Yes?
Do you think you would do a different answer without, you didn't allow for the homologous recombination, instead did it through a different mechanism, like an insertion?
Yeah, so the question is whether if we used a different mechanism, we would get a different result. And the reality is I have no idea. I mean, I can speculate, but I have no data behind it, so it would be pure speculation. My thought is that, my bond is going off, so I need to stop now.